Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on module Lipids Oxidation Part 4 from the paper Lipid Metabolism. The objective of this module includes to understand the unsaturated fatty acid oxidation, the alpha and omega oxidation process, and how the fatty acid oxidation takes place in peroxisomes. First of all, let us see how the unsaturated fatty oxidation takes place. For the unsaturated fatty acid degradation, two additional steps are required along with the other reactions of beta oxidation catalyzed by enzymes, an isomerase and a reductase. The monounsaturated fatty acids requires only isomerase, while the polyunsaturated fatty acid requires both isomerase as well as reductase. The concept map of the metabolism of fatty acid. Again, if we look at the overall process of the fatty acid metabolism, it is allied or interlinked with several other metabolic pathways, including the carbohydrate metabolic pathway, the amino acid metabolic pathway, the steroid metabolic pathway, as well as its relationship with the intake of the dietary lipids. The dietary fats or the lipids, after esterification and its conversion to triacyl glycerol, undergo lipolysis to form the fatty acid molecules. The breakdown of fats to the fatty acid as well as glycerol results in the reactions for the oxidation of fatty acid through beta oxidation, through alpha oxidation, through omega oxidation. The ultimate aim of these oxidation to yield acetyl-CoA, and this acetyl-CoA is being utilized through lipogenesis process to form again the fatty acid molecule, which again recycled through esterification to triacyl glycerol and further break down to fatty acid and acetyl-CoA. Acetyl-CoA can be considered as a master regulator of most of the metabolic pathway in the cells, whether it is carbohydrate, amino acid or lipids. The so formed acetyl-CoA undergoes cholesterologenesis to form cholesterol, which further results in the formation of steroids through steroidogenesis. Acetyl-CoA not only forms cholesterol and steroids, but during starvation also results in the formation of ketone bodies through a process called as ketogenesis. Not only the breakdown or the synthesis of the cholesterol and ketone bodies, but also this acetyl-CoA enters to the Krebs cycle or the tricarboxylic acid cycle or the citric acid cycle to yield energy, to provide energy to the cells, and ultimately results in the formation of carbon dioxide and water. The problems associated with the monounsaturated fatty acids. For example, the degradation of palmitoleiate or 16 carbon unsaturated fatty acids having the double bond between carbon 9 and carbon 10. The activation and the transportation occurs similar to that of saturated fatty acid, that is, across the inner mitochondrial membrane. The first two reactions are the same, while the third reaction does not occur, as cis-enoyl CoA is formed instead of trans-enoyl CoA. Therefore, the enzyme is not specific. Another problem associated with the formation of double bond between carbon second and third is prevented by the already present double bond between carbon third and fourth. This problem is solved by shifting the position and configuration of the cis double bond into the trans double bond by isomerase enzyme. The oxidation of a monounsaturated fatty acyl CoA, which is oleail CoA. This oleail CoA, through the beta oxidation, after the completion of 3 cycles, form cis-do-decanoyl CoA, which is a 12 carbon compound. Along with it, 3-acetyl CoA is released. This 12 carbon compound cis-do-decanoyl CoA, by the catalytic action of enoyl CoA isomerase, gets converted to trans-do-decanoyl CoA. This trans-do-decanoyl CoA is acted upon by enoyl CoA hydratase to form L-beta hydroxydecanoyl CoA, which is also a 12 carbon compound. Further, the L-beta hydroxydecanoyl CoA undergoes 5 cycles of beta oxidation to form 6-acetyl CoA. What are the problems associated with polyunsaturated fatty acids or PUFAs? For example, linoleate, the carbon 18 polyunsaturated fatty acid with cis 9 and cis 12 double bonds. The cis 3 double bond formed after the 3 cycles of oxidation is converted by isomerases to a trans double bond. But another round results into a cis double bond and its dehydrogenation yields a 2,4 dienoyl intermediate. By the catalytic action of acyl coenzyme A dehydrogenase. This intermediate is not a substrate for the next enzyme in the beta oxidation pathway. This deadlock is overcome by the reduction of this intermediate to a trans 3-enoyl coenzyme A by NAPGA dependent 2,4 dienoyl CoA reductase enzyme. The product trans-enoyl coenzyme A is converted to trans 2 form by cis 3-enoyl CoA isomerase. The remaining steps are the same. The oxidation of polyunsaturated fatty acids, linoleate, this 18 carbon compound, linoleal CoA, undergo 3 cycles of beta oxidation to produce cis 3 and cis C linoleal CoA compound, which through isomerization gets converted to trans 2 and cis 6 compound by the catalytic action of enoyl CoA isomerase. This compound further undergo beta oxidation one cycle only, and the first oxidation of the second cycle to release acetyl CoA, ultimately forming the trans 2 and cis 4 compound, which utilizes NADPH and proton in presence of 2,4-dienoyl-CoA reductase to form trans-3-dienoyl-CoA. This trans-enoyl-CoA undergo isomerization to form trans-2-enoyl-CoA, which undergo beta oxidation of 4 cycles to release 5-acetyl-CoA. Alpha oxidation. Relatively unimportant pathway for the production of energy and are mainly used when dietary fatty acids are branched or chain and are methylated. For example, plants contain phytol, and these phytols are present as one of the constituent of the chlorophyll, the long hydrocarbon side chain, and its metabolite, which is known as phytanic acid, is metabolized by alpha oxidation. The phytanic acid is present in milk lipid, food derived from milk, and animal fat, and is metabolized by initial alpha hydroxylation followed by dehydrogenation and decarboxylation. The phytanic acid contains a methyl group attached to the beta carbon, suggesting the fact that the compound is not a substrate for beta oxidation. Hence, the phytanic acid must undergo alpha oxidation. This pathway is also operative in brain and plant tissues as well. The oxidation of phytanic acid. Phytanic acid is a branch chain fatty acid originated or derived from phytol. This type of oxidation is called as alpha oxidation, as the conversion takes place at alpha carbon instead of beta carbon like other fatty acids. In the first step of the reaction, phytanic acid gets converted to phytanoyl-CoA, which further through hydrogenase reactions form 2-hydroxyphytanoyl-CoA. Subsequent transformation results in the formation of prestanol and prestanic acid under the influence of dehydrogenase enzyme. Then this prestanic acid undergo multiple rounds of beta oxidation to form medium chain fatty acid, and finally this medium chain fatty acid undergo mitochondrial beta oxidation to yield carbon dioxide and water. The initial hydroxylation reaction is catalyzed by a mitochondrial enzyme monooxygenase that requires oxygen, magnesium ions, NADPH, and a heat stable cofactor. Conversion of alpha hydroxy fatty acid to carbon dioxide and the next lower unsubstituted acid appears to occur in the endoplasmic reticulum and to require oxygen, iron, and ascorbate. The salient features of alpha oxidation are as under. The first salient feature is only the free long chain fatty acids can serve as a substrate. The next feature include the involvement of molecular oxygen indirectly. It does not require coenzyme A intermediates, and also it does not lead to the generation of high energy phosphates. Oxidation. Omega oxidation is a minor pathway in which omega terminus carbon is oxidized to the carboxyl group to form dicarboxylic acids. Further metabolism takes place as it happens in beta oxidation process. The biological oxidation of fatty acids at the omega carbon atom was first reported by Verkade and his group who isolated the dicarboxylic acids of the same chain length from the urine as those that were fed in form of triglycerides. So he proposed, based on his experiments, that certain acids were first oxidized at the omega carbon atom and then further metabolized by the beta oxidation process, proceeding from the both end of the dicarboxylic acid. In this diagrammatic representation, we can see how the bacterial as well as animal microsomes are directly or indirectly related to the formation of the oxidation of the fatty acids as well as utilization of flavoprotein and NADPH along with reduction of non-heme iron protein, rubidoxin, as well as the action of hydrolases. Both the NADPH and flavoprotein in an oxidoreductase reaction or the redox reaction form a specific non-heme iron protein in animal microsome and rubidoxin a specific NHI protein in the bacterial system. These non-heme iron protein with the action of cytochrome P450 and by the utilization of the oxygen ultimately results in the formation of acetaldehyde and acetic acid, carboxylic acid through the reduction of NAD+. The mechanism involves an initial hydroxylation of the terminal methyl group to a primary alcohol. In animals, the cytochrome P450 system is the hydrolase responsible for this alkane hydroxylation, whereas in bacteria, rubidoxin is the intermediate electron carrier which feeds electron to omega hydroxylase system. Summarily, the methyl group is converted to a CH2OH group which subsequently oxidizes to carboxylic acid, thus forming dicarboxylic acid. Once formed, the dicarboxylic acid may be shortened from either end of the molecule by the beta oxidation sequence to form acetyl-CoA. The series of reactions as depicted in the picture now assumed to have an extremely important scavenging role in the bacterial biodegradation of both detergents derived from the fatty acid and, even more important, the large amounts of oil spilled over the ocean surface. The rate of bacterial oxidation of floating oil under aerobic conditions is estimated as high as 0.5 gram per day per square meter of the oil surface. The bacterial oxidation of oils is brought about primarily by omega oxidation mechanism. Fatty acid oxidation in peroxisomes. Few oxidations occur in cellular peroxisomes, and the peroxisomes are having high concentration of catalase enzyme. It is important for the oxidation of octanoyl-CoA, and it helps in the shortening of long chain for making better substrate of beta oxidation in the mitochondria. Flavoprotein dehydrogenase present in peroxisomes transfers electrons to yield oxygen and finally hydrogen peroxide instead of capturing the electron as FADH2 as occur in the mitochondrial beta oxidation. So what triggers the switch towards fatty acid oxidation? During the fasting, the ratio of insulin and glucagon is low, resulting in the stimulation of the lipolysis process. The overall of the fasting or the stress related leading to opposite regulation of the insulin is the switch from a fuel economy based on carbohydrate, in which the major proportion of the energy comes from lipid oxidation. So students, in this lecture we have learned about the alpha oxidation and the omega oxidation process. The fatty acid oxidation occurs in peroxisomes, and also what triggers the switch towards the fatty oxidation process for the complete metabolic activity of the cell. Thank you.